Okay, cytoskeleton. Good. So I've drawn them all in black, all these little black lines, parts of the cytoskeleton. With the photographs or images that I'll show you, it extends a lot more than what I've drawn it, okay? And there are different components of the cytoskeleton that we'll talk about individually, and I've not sort of distinguished these drawings, these parts of the cytoskeleton into those different parts. The only thing I will do is with these, I'm going to put little vesicles on them. Okay. All right, so cytoskeleton is this network of fibers that extends through the cytoplasm. Now, there are a lot of different roles of the cytoskeleton, all right? But it organizes the cell structure, helps organize the activities, helps maintain cell structure, and helps anchor many organelles in place. All right? So, and as we look at each of the different kinds, you'll get a better picture about what it does. So, there are three different molecular structures, or three different components to the cytoskeleton. You've got structures called microtubules. You've got structures called microfilaments. And then you've got structures called intermediate filaments. Right? And we're going to look at each one of these separately. Right. So we'll look at what they're made of and what their function is separately, each one of these. Okay? All right. So here's an epifluorescence image showing at least two parts of the cytoskeleton. The green little lines you can see, they're microtubules. So they're tagged with a green dye, a green stain. Red is microfilaments. Oh, what's the third part of the cytoskeleton? Intermediate filaments, good. They're not shown in this image, just two parts, microtubules and microfilaments. And have a look at how dense they are, different locations, very densely packed filaments and fibers. And this blue part is the nucleus of the cell. Okay? So can you see?
Usually surround the nucleus like that, or they all?
It does. Some parts do. And we'll talk a little bit about that. Intermediate filaments do. Okay? In fact, they almost form like a cage around the nucleus. All right. So first part is the microtubules. We want to talk about those. Now, microtubules, as the name implies, are small tubes. They're hollow tubes made of the tubulin subunits. So they're made of protein. And they have a lot of varied roles in the cell. One of their functions is that they help maintain the shape of the cell. Remember, animal cells don't have a cell wall, right? They help maintain the shape of the cell. Now, this particular part is compression resisting. All right? Now, not to get too much into structure, but I want you to think about this. Sorry?
Like shock absorber?
Well, it's a bit like, think about it as kind of like concrete. Concrete is really hard to compress, right? But concrete, if you stretch it, it breaks easily. You can snap it easily. Yeah? So these are really sort of compression resisting. Other parts of the cytoskeleton are tension bearing, right? It's why you put rebar inside concrete, steel bar. The steel is very good at resisting tension. Concrete is very good at resisting compression. The two together, really strong. Okay. All right, so they help maintain the shape by resisting compression. They have a role in cell movement. Cells have, not all cells, depends on the cell, can have structures called cilia and flagella. And I'll elaborate on those in a moment. They can have cilia and flagella can have a role in moving the cell, propelling the cell. Or sometimes they have a role in moving fluid over a surface. All right? And again, I'll elaborate on that. Chromosome movement in cell division. Chromosomes move in cell division, and the microtubules have a very important role in chromosome movement during cell division. Now, do you remember what chromosomes are? It's folded and coiled, very highly packaged DNA. And you can see chromosomes through the microscope. We're going to have a lab and you'll look at chromosomes through the microscope. They also are tracks for organelle movement. They're the track system. Think of them almost like a monorail throughout the cell. Organelles move along them.
So in that video, that protein that was walking that, he was walking on the cytoskeleton.
He was walking along,
Or
yeah, was dragging that vesicle along a microtubule.
Okay.
We're going to watch that video again once we've finished talking about the cell, and you're going to understand all of it, I'll tell you. All right. So have a look at this epifluorescent micrograph. All those green fibers, they're microtubules. All right, you can see very extensive system throughout the cell. And here's what they look like up close. This is a diagram. They're tubes. They're made of little tubulin subunits that sort of wrap and coil themselves into these tubes.
What happens inside?
I don't know what's inside. There'll be other molecules, most likely water. Okay? All right, so cilia and flagella then. All right, this micrograph on the bottom left shows cilia. So this shows the surface of a whole bunch of cells, actually cells that line your trachea. Right? Those cells that line your trachea have lots of cilia on their surface. And cilia beat, usually in one direction. They're able to beat easily in one direction. Okay? And the cilia and the cells that line your trachea beat in one direction to move liquid from your lungs up to the back of your throat so you can swallow it. Because, of course, when you breathe, there's a whole bunch of junk in the air you breathe, dust and pollen and whatnot. That can get trapped on mucus in your lungs, and that all needs to be moved. And it gets moved to the back of the throat so you can swallow it, get rid of it. So this is what cilia look like. And then flagella, singular flagellum, they sort of move in this sort of snake-like motion, and they help propel cells.
Like a snake or a whip.
Kinda, in a snake-like movement. I'll elaborate on how each of them moves in a moment. But cilia and flagella are these little structures off the cell which move, and the microtubules are present along their length, and they help them move. So let's add some cilia and flagella. All right, they're not membrane, so I'm going to draw them in black. So I'm going to add a whole bunch of cilia, say, over here. Now, cilia they may not occur in a little patch like this. In fact, cilia might cover the entire surface of the cell, or they might just be present on part of the cell. All right? But I'm only going to draw them on a little area. Now, we're kind of making a cell that doesn't really exist, right? But now I'm going to add a flagellum to this cell, a single flagellum. All right, a flagellum. So cilia are usually much shorter than the cell that they're attached to, and usually much shorter than a flagellum. There's usually many cilia present on a cell, whereas the flagellum, there might only be one or maybe a few. Okay? So the differences between cilia and flagella: shorter than the cell, much smaller than the cell, and many of them per cell. Flagellum, usually much longer than the cell. There's only one or a few per cell. All right? Okay. And along the length of the flagellum and along the length of the cilia, there are microtubules inside. Now, flagella and cilia sort of move slightly differently. So think about a flagellum that's attached to a sperm cell, and I'll show you a video of this. It sort of undulates like this. Okay? The flagellum does. Yep, that's why I've kind of drawn it like that. Yep. The cilia, on the other hand, moves a bit differently. It's a bit like an oar. Solid stroke, and then it relaxes. Solid stroke, relaxes. Everyone okay with that? Yep. And it's obviously that pattern of movement suits their function. The flagellum, in the case of a sperm, has to propel it in a straight line through whatever liquid it's got to swim through. This is moving fluid over the surface. Okay. All right. So here's a little video clip showing sperm swimming. Poor little guys aren't going anywhere.
I know, I feel kind of bad.
Yeah. All right. And this little video clip shows a paramecium, it's a little protist. Its entire surface is coated in cilia. It's hard to see the cilia in this movie, but you'll see that it enables the organism to move.
Cilia's moving a lot faster than flagella fellas.
So do you remember seeing cilia on Stentor in the lab, the one that sort of looked a bit like a wine glass? They were just present on that rim and they moved liquid over its surface. Now, paramecium usually move much faster than that. They're like little jet-propelled. They're not jet-propelled, they move really fast. But sometimes you can slow them down. You can make the media they're in very viscous. It's like trying to make them swim through honey, and that slows them down. So if you were to cut through a cilia or a flagellum, this is sort of the arrangement of microtubules that you'll find. It's quite complicated, yet fairly simple in some ways. All right? So I'm going to play this video clip, and you'll see sort of how that arrangement enables the cilia and flagella to work.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella, or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
Good. Now, did you catch something she said? They propel sperm in many animals and many plants. There are some plants that make sperm, not pollen, that have a flagellum that can swim. Anybody know what a ginkgo tree is? Yeah? They make a herbal preparation for it. It's meant to help with your memory. Ginkgo biloba. It's got these very characteristic shaped leaves that kind of look like this. And these leaves are present in the fossil record, so this tree has remained unchanged for millions of years. But there's separate sexes, male and female plants. They don't make flowers, okay? And the male makes these motile sperm that have to swim where they're produced, down the trunk of the tree, through the soil, up the trunk of a female tree, and then to fertilize the fruits on the female tree. Isn't that kind of cool? And the female fruits, when they make fruits, they're about as big as a Malteser. Malt ball. A malt ball, right? But when they're mature, they smell terrible. They smell a bit like dog shit, right? So people don't like to have the female trees around, right? Because in the fall, when all the fruits drop, it's like 1,000 dogs have crapped on your front lawn. All right. So now we're going to look at the role of microtubules in moving chromosomes. All right. So this is an amazing movie, and when I found this, I was actually really excited because it was shot by a guy called Andrew Beyer back in, I think, the 1950s on 16mm film. And when I was a grad student, it was a long time ago, I was a grad student at the University of Oregon, and Andrew Beyer was a professor there. He was this old guy, sort of a quintessential scientist. He had glasses, and I kid you not, there was tape in the middle, right? And he had pens with a little pen protector in his pocket, just quintessential. And he had a lab down the way. And I didn't know him or any of his work. You know, I was 21 when I started grad school, but that was really cool when I found this moving. He did some of this work. So you're going to see chromosomes moving. You're not going to see the microtubules, but the microtubules have a role in moving these chromosomes. So these are chromosomes that have just formed inside a nucleus of a cell. And now they're being moved around by microtubules that you can't see. And you're going to see each chromosome is going to get pulled in half. Half goes to one side, half goes to the other.
The cilia pulling them in half?
Microtubules, not cilia, but microtubules within the cell.
So this is... Okay,
This is during cell division.
that like mitosis.
This is mitosis, yeah? So you notice now the chromosomes have gone away, the DNA is decondensed, and this would be the cell wall that divides one cell from the new cell. All right? So there's no cilia or flagella involved, but microtubules run the length of the cilia and flagella. Okay? But another role is that they move chromosomes, and that you saw. Okay, so another role of microtubules is they act as tracks for organelles to move along. Yeah? So this is what an electron micrograph would look like. There's your microtubule. This is a vesicle, two of them being pulled along that track. It's like a monorail. And up here's a diagram, kind of what it looks like. There's your microtubule, there's your vesicle, there's the motor protein. It takes ATP energy to change the shape and drag it. Yeah.
So are all the vesicles, like, that's the only way they move is on...
I wouldn't say it's the only way they move, but it's a major way that they move. And they move quickly. They can move quickly along the microtubules. Okay? All right. Good? Now you're going to watch a movie, all right, of how quickly these organelles can move in vitro. That means in a test tube or in a petri dish, not in an organism. See the organelles moving along the microtubules? It's crazy, isn't it? It's fast.
So they move too, like the microtubules?
Yeah, everything's dynamic. The microtubular system of the cell is quite dynamic. It gets built up and torn down and moved around in a very dynamic way.
So it looks like it's just nonsense, but there's a point for all that.
Oh, crikey, yeah.
That's great.
Yeah. So here's another one, transport of vesicles along microtubules. So here's a cell, that's the nucleus. Here you can see microtubules. It's really hard to do this kind of microscopy. Okay. All right. So now we're going to look at the second part of the cytoskeleton, which would be the what? Microfilaments. So microfilaments, they're not tubes, they're filaments. In fact, it's two twisted rods of actin. Actin is a protein, it's a contractile protein. Two twisted rods of actin. Okay. And this micrograph down here, you can see that they've used a red fluorescent dye to tag all the microfilaments in a cell. And you can see how extensive that microfilament network is. And this one over here shows you those two rods of actin sort of twisted, a bit like a cable or a rope. And here you can see these actin subunits that make up this. All right. So, microfilaments then help maintain cell shape. These are the tension-bearing ones. All right. They're very strong in tension. You can't break them if you pull them. All right. So they help maintain the cell shape. They're very important in muscle contraction. They have a role in something called cytoplasmic streaming, which we've already seen in lab, but I'll show you a video of it. And they have a role in the movement of amoeba, causing amoeba to move. Right, again, we've seen that in lab, but I'll show you a video. And they also have a role in the division of animal cells. I'll show you that last one, division of animal cells, right now, since I don't have a movie for it. But if you think about an animal cell, does that. There's an animal cell. And for an animal cell to divide, it kind of does this. It forms these little pinches in at the middle. And then it pinches in a little bit more. And then it pinches in a little bit more until it pinched off into two separate cells. that pinching in is caused by microfilaments contracting. Okay? So everything that microfilaments do comes down to their ability to contract. Pretty much, except the tension bearing, really. They're very, very strong. So this micrograph shows the microfilaments part of the cytoskeleton throughout these cells. This is the lining of your, I think it's the lining of your gut. All right, and you can see, very extensive network. Yep, gives it strength. So they have a role in muscle contraction. These are the actin filaments, the ones that are in orange. There you've got some myosin filaments, which is another protein found in muscles. And what happens is they kind of move over each other like this. As a result of the actin filaments moving kind of like this close, then the whole muscle cell gets shorter and the whole muscle contracts. So when you start to work out, you're going to increase that part of the cytoskeleton of muscles massively. Right? That's why your muscles get bigger, get stronger. All right, so look at cytoplasmic streaming then. See the chloroplast motoring around? Yeah. The cytoplasm is moving as a result of those microfilaments contracting. And the chloroplasts are kind of moving around in that flow. So this is amoeboid movement. So amoeba are kind of cool. They've got something called plasma cell, sol, and plasma gel. So on the inside, they're more gel-like. On the outside, they're more liquid-like towards the outside. They do kind of have a back end and more of a front end. And what they do is they squeeze part of the cell, and then they make these projections called pseudopodia. But that squeezing of the cell is done by microfilaments. It gets squeezed out of one part into another part.
Like a piece of clay.
Sorry?
Like a piece of clay, you're stretching that.
Yeah, kind of. It's almost like filling up a glove with water or a balloon. You squeeze one part and it extends another part. That's a pseudopodium, and you can see it's all this cytoplasm is being squeezed into that pseudopodium. And then maybe it changes its mind. This is sped up a little bit. Or not at all. it should be playing. I don't know why it's not playing. Well, I shot this video and I made it, so I'm a bit bummed it's not playing. Oh, there you go. Oh! Didn't show you the sped up, but what happened was after I was shooting it, is the cell lysed, it burst. Water evaporated, cover slip got with it, and it burst.
So do those contain a contractile vacuole then?
They do. Amoeba do, yeah.
Wait, can you play it again?
Sorry?
Can you try playing it again?
Yeah, let's see. Oh. Oh, it started to behave, then not. You'll see the first couple of seconds of the sped up. It's worth seeing it burst. Pop. But it's still going, even though it's kind of burst or become flattened. Still squeezing. Or maybe it didn't burst here. Maybe the cover slip just got like squashed it really flat. I think that's perhaps what's going on. Right. See this little food vacuole there? Food vacuole, feeding on these guys. Yep. All right, so let's have a look at this next component of the cytoskeleton, and that's intermediate filaments. All right. So they're called intermediate filaments because they're filaments, they're not tubes. But size-wise, they're kind of in between the microtubules and the microfilaments. Microfilaments are very thin, microtubules are kind of thick, and these filaments are size-wise in between the two. And these are made of fibrous proteins, a whole different mishmash of different fibrous proteins, but one major component, one major protein is keratin, that your hair and nails are made of. And that's coiled into these cables. All right, So let's have a look at the intermediate filaments, what they do then. They support cell shape. They are extremely strong tension bearing. So nerve axons, your nerve cells, you've got some very long nerve cells in your body, right, that stretch from the base of your spine to the end of your toe. Think about when you sort of extend your leg, they stretch. You don't want them to break, right? Nervous tissue is remarkably strong. Yeah, you seen that movie, what was it, 72 Hours? Where the guy stuck down the hole, his arm was stuck.
Oh, yeah.
Yeah? And he had to cut his arm off. He had to break the bone, sever his arm. So the nerve was one of the toughest parts to cut through. Nervous tissue is very, very tough. It's very strong, thank goodness. So nerve axons have quite a complex intermediate filament part of the cytoskeleton to make them strong. Your skin is remarkably tough. Has anybody ever had to suture human skin?
Had to cut human skin?
Suture it.
What's suture?
Put a stitch in it.
Oh, no, I don't think so.
No? Some people work in the medical field. You might have done it. You're in the
stitches.
military. It's really hard to get that needle through human skin.
Is that
Human
why it's
skin,
hooked?
well, it makes it easier to get it in.
So you're not going through tissue.
Yeah.
If it's not hooked, you end up going through the actual muscle.
It's just tough. And I know we think we get scratched easily, but it's remarkably tough, right? Human skin. All right, so bear tension. That's why your skin is strong. It fixes organelles in position. So many of the organelles in your cell are fixed in their position. They don't move much. Now, mitochondria can. They kind of like move a little bit throughout the cell. But the nucleus is kind of fixed in place. And there's almost like a cage of intermediate filaments around the nucleus holding it in place. Because remember, there's going to be flow, directionality of things. And they need to be held in the right place. Think about it. Every time you jumped up and down, if everything just got shook up like a can of vegetable soup, that wouldn't be good. Everything has to be held into place. And this part of the cytoskeleton is a lot more permanent than the other parts. The other parts get assembled and disassembled, made up, broken down. Think about muscle cells. They get bigger and smaller with use. And microtubules are continually being made up and broken down depending on what you need. But this is a lot more permanent part. So this table is in your textbook. It's an excellent table. It summarizes, compare and contrast those three components of the cytoskeleton. I recommend you make your own similar tables. All right? It's a really, really good study tool and study aid. Okay, so next time we'll start off with the extracellular matrix, and everybody have a fantastic spring break.